My name is Tim Noon. I'm a project manager for the U.S. Army Corps out of the Buffalo District. I'm supported by a pretty diverse team that's represented by some of the logos that you see on the screen there. Today, you're going to hear from Shaylin Kaufman, who works with our Engineer Research Development Center as a research geologist. You're going to hear from Chad Toussaint, who's a physical scientist from the U.S. Geological Survey. You'll hear from Derek Schlee, a senior ecological engineer at our partners at Limnotech. And again, we'll be taking questions as a team as we go. So I'll drive forward here. Just to frame up the problem and what we're looking to do out here, right, is that we all have an understanding that harmful algal blooms can be driven by excess nutrient loading, particularly phosphorus, into the Great Lakes. And our project is a project that we're undertaking as part of the Great Lakes Restoration Initiative to look at reducing nutrient loading in Great Lakes tributaries through the concept of phosphorus optimal wetlands. And we'll talk a little bit about what that is. We want to share results with you today about what we've been finding in our work out there in Defiance, Ohio. We also want to build opportunities to foster some collaborative research as we move forward with interested stakeholders in the area. The agenda for today's webinar is that I'll go through a quick background on the project. We'll talk about wetlands and phosphorus optimization as a concept. We'll talk about the specific monitoring and the operational research that we're doing out there in Defiance. We'll talk about the results to date, and then we'll talk about the next steps and where we go from there. One more slide here just to talk about framing the problem and what we're looking to do with it. This is a demonstration project, right? So our goals is really to work within the GLRI Focus Area 3, to optimize phosphorus retention on the landscape, and to demonstrate a new approach to how wetlands are restored and can be managed to ultimately bring nutrient loading down, right, through the practice of wetland restoration or P optimization. It's a relatively novel approach, but obviously wetland restoration is something that occurs across the basin. So there's really great opportunity to share what we're learning with wetland practitioners across the Great Lakes. Just a slide here, and I apologize if this is maybe typical of the Army Corps to make flow charts here for you, but just to kind of give a quick overview of how this project is broken out in terms of who's all involved. We are working, as I mentioned, I work for the Army Corps Buffalo District. We're working in collaboration, though, with our partners at the Engineer Research Development Center, or ERDIC, as we sometimes call it. That's our research lab that's ultimately driving the operational research out there. We're doing this in partnership with the U.S. Geological Survey and also through contract with our partners at Limnotech, who are on the line with us as well. I'll speak briefly to this timeline slide, and I'll lean on Derek Schlee, who's with me here, to see if he's got anything to add to it. But the bottom line here is the work that we're doing in Defiance for a demonstration wetland comes from a long period of engineering and design, and before that, a study phase to really site select and ultimately come up with a concept for phosphorus optimization. So here you'll see kind of the timeline of where we came from July 2020, where we took concepts and moved it all the way out to an implemented and fully operational wetland in 2022 that we're still doing work on out there for another year. We're out there until May of 2025 with our current phase of work. And all of this work builds on some significant research and a prior study phase that happened to really come up with the concept of phosphorus optimization before that. I'll pause here. Derek, anything that you wanted to add to this slide? I know you contributed here as well.
No, that's good. I'll just correct. I think we have 2026. Yep. So another year
Yep.
of the operation before this project phase ends. Yep.
Yeah, thanks, Derek. All right, and with that, I'm going to turn it over to Shaylin to take these next steps.
Yeah, absolutely. And so where I kind of want to start here, right, we, like, as Tim said, right, we know that wetlands are really effective for managing nutrients. And to highlight also, another thing Tim said was building upon the body of work that's already been, right? So you can look here at this bar chart, and this is cumulative load reductions, and then these are 3 individual wetlands and their cells. This work comes out of Lemke et al. 2021. This was a 12-year study where they evaluated the edge of field wetlands for their efficacy for nitrogen and phosphorus removal. The big thing here that's visible is that this really led to a roughly 50 to 80% reduction in phosphorus from inflow to outflow. And so one of the things is that, myself, everyone on this call really is just deeply integrated with trying to create best management practices. And a lot of implementation has already happened, and we just haven't necessarily met all of the regional targets. And so we have a really cool opportunity, right? Of figuring out how we can use these wetlands to best reduce our nutrient loads. They act as natural filters. They help us retain P by trapping it and binding it within the soil. There's a load of biotic processes that also help us really, get some of that moving. And so the other thing is that wetlands are great because they allow us to really have a host of co-benefits. So whether that's habitat, biodiversity, recreation, integration within the community. And so we've got a couple of challenges, though, to keep in mind. And so this is kind of where we start to get into the goals of the project. Many ideal sites might not have a real capacity for storing additional phosphorus. In fact, some of them might be hotspots for legacy phosphorus. And so we want to keep that in mind as we're selecting sites. And even in sites where our phosphorus capacity is good and it can increase, we might end up, not having as much as we thought, or not necessarily understanding how best to manage it for long term. And so within that is this limited life cycle analysis that we've not really seen so far. How do these sites perform over 5 years, over 10 years, over 15 years? And so our goals here are really just to guide and develop operational guidance for the when, where, what, how of these nutrient reduction wetlands, and then really getting started on this process of developing solutions that can be scaled up and implemented at this region-sized problem within the Great Lakes. And so as we kind of transition then to this next slide, this research has been a series of scales really from the smallest to this larger demonstration scale size. And so we have taken the time to do benchtop and mesocosm studies, and we use those to inform operations, really optimize the design features, and think about what long-term management is going to look like for greatest return on investment. And so we're not going to get into all of them, but I've picked out a couple we can highlight. Across the breadth of these, we've looked at temperature and oxygenation and, hydro patterns and residence times and amendments. The first one here is residence time. And so on this graph, what you can see is that the y-axis is cumulative rate of P removal. And then along this bottom axis is time. And so there's 3 curves that you can see. These are the 3 operational scenarios we tested with residence time. The top one in light gray is bare soil. The middle one is the established vegetation. And then this bottom curve is vegetation that's been established plus the use of an amendment. And so what we did was we allowed our mesocosms to just hold water at a known concentration of phosphorus. And then we tracked at what point this plateaued off, right? Or when we had hit the maximum phosphorus removed from the water, rate-wise. And so within this, we really found that, if you have bare soil, it is highly effective, and we'll show that on the next slide as well. But it does take 12 hours, right, to hit that maximum rate of P removal out of the water. And as we add in a more natural environment, something like vegetation, where you go down to 8 hours, and when we add in amendments, we're down to 4, right? And so operationally for this means for us that, as long as we are, as we're pumping water into our system, as long as it's in there for 8 hours, front to back, we are really just going to get the most bang for our buck with these pumping events. And so I've just got two more examples of this. On the next slide here, so on this next slide, we see hydro patterns. And so I'll explain this graph really quick. On the y-axis, this is phosphorus retention over 24 hours. And so on the bottom axis here, what we have are 5 operational scenarios. Each operational scenario has 1 bar with the circles for bare soil, and then 1 bar with the crosshatches for vegetated or established vegetation. The other thing to notice is at the top of the bars, those are removal efficiencies. So that's just front to back how much phosphorus we got out of our water. And then along the bottom, you can see the amount of water that we cleaned during the time. And so one of the keys here is that if you look at the 1 pump scenario versus the 2 pump, and to be explicit, that's 1 pump is kind of our low flow rate versus 2 pumps is our higher flow rate. You can see that, over that same period of time, we've treated double the water. And not only that, we really didn't lose much in efficiency, right? Those are only 1 or 2% different overall. And so that is good for us to think about operationally because, maybe we care more about the quantity of water we're able to clean over the same amount of time than we care about, necessarily maybe we're okay with, right, like a slightly lower efficiency. The other things to highlight here are that if you look at 1 pump versus this 2 pump 2-hour hole, that's a pulsated flow. So we're flowing for some time and then holding for 20, and this cycle kind of repeats for 24 hours. What you'll see is that if we're looking specifically at the vegetated scenarios, we are cleaning the same amount of water in the same amount of time, except for when we are allowing this hold, we are actually increasing the efficiency by nearly 30%. And so unlike when we have the high flow versus low flow rates, when we have the, 1 flow static pumping at 480 gallons versus this 2 pump 2 hours one, same amount of water, but we really increase the efficiency. And if we look just directly at the bars, as we kind of go across this and we have these increased holding times, what you can see is that, versus the high flow versus, let's say, 2 pumps 2 hours, we're cleaning 4 times as much water when we have just steady high flow. But, we really are not necessarily taking in as much phosphorus retained. And so when we have this 2 pump 2 hours, we're still getting a lot of phosphorus off of that, even though it's so much less water. I'll transition us really quick. This is just a side note. So we don't have the data up on this currently, but as another guide for how these operational scenarios can affect what we decide to do, we ran experiments where we looked at different vegetation management strategies. And the big takeaway that I just want to say quickly is that, when we allow the vegetation to senesce, or maybe we harvest it and we gather it for baling or something like this, we are at the end of that, as we let the water flow back over the surface, over 2 to 3 weeks, we're still losing less phosphorus, significantly less, 5 to 7 times less than when we just run the system at defiance 1 time. So these are all, I think a real point here is that these aren't cut and dry, and these operational scenarios just really help us guide how we should be implementing these things just based on our operational goals. And with that, I will let Derek tell you about how defiance works.
I apologize for not advancing some of the animations here for you, Shaylin.
No, that's
also,
okay. That's okay.
Yeah, WebEx has a tendency to auto advance, so I'm controlling that for that here. But yeah, over to Derek. Thanks, Shaylin.
Yeah. Okay. Thanks, Shaylin and Tim. So moving from the mesocosm studies that Shaylin just described back to the field demonstration site in Defiance, Ohio, I'll use this slide to talk through how the wetland actually functions to treat runoff from Colwell Creek, which is shown at the top of the screen there in the lighter blue. And that creek drains about 900 acres of primarily farmland before emptying into the Auglaize River, which is a tributary of the Maumee River. So the wetland treatment system begins with a settling pond that diverts a portion of the creek flow during elevated flow events. And from there, we have a pumping system that lifts water into the first of 4 consecutive wetland cells. And so as you'll see in the dark blue arrows, which demonstrate the flow path as water moves through the wetland through each of these 4 wetland cells, we have some separation between the cells with these berms, and then also the use of earthen spillways and agri-drains that convey the water between the cells. So as the flow paths follow this meandering, slightly deeper channel, they also have the opportunity to spread out on a bench or floodplain area as more and more water fills the wetland. And then finally, we get to the end of the wetland at what we call cell 4, where water exits through, again, both an overflow spillway and an agri-drain, and then discharges into this drainage ditch, which then empties back into Colwell Creek. Go ahead to the next slide. A quick look at some of the monitoring features with this schematic that demonstrates the various devices that have been deployed to accomplish the research objectives of the demonstration site. Chad will go through the USGS monitoring equipment shortly, but those are indicated by the green diamonds on the schematic here. Limnotech has installed water level sensors, water quality sondes, online nutrient analyzers, and automated ISCO samplers at key features within the wetlands, really to provide insights at how it's performing and improving water quality. And I'll wait till later to describe our monitoring equipment in a little more detail when I get into some of the results. But for now, I'll hand it off to Chad to describe the USGS work.
Hello, for those who've met before, nice to talk with you again. For those I'm speaking with for the first time, I'm Chad Toussaint, physical scientist and project manager for the U.S. Geological Survey in Columbus, Ohio. As Derek said, I'm going to highlight some of the contributions from the USGS to this project, and that's mostly looking at the flow monitoring and making information publicly available and looking at some of the, as Shaylin had highlighted, some of the biodiversity impacts. Next slide. The role of the USGS in this project from the beginning is to determine the mass balance equation. We look at what's entering the system, how much is entering the system, and what and how much leaves the system. The USGS can provide the big numbers, but thanks to the other monitoring partners at Limnotech and ERDIC, internal mechanisms of the wetland will be revealed in their study work, or in Shaylin's case already. Over the next couple of slides, I'll present the ins and outs of the wetland. Next slide. Our monitoring starts before the wetland. We start by monitoring process in Colwell Creek, click, to get a baseline data for the discharge from the drainage area that Derek described, and to assess the nutrient capacity, click. By collecting these data, we can assess not just the wetland component, but the holding pond. I want to draw your attention to the Hydrologic Imagery Visualization Information System, or HIVIS. Click. This is a publicly available system that allows you to view real-time data and photographs. After water passes through the culvert back to the open channel at the first gauge, it's diverted into the wetland. Click and click, and then pumped into the start of the wetland. Click. This is our second sampling spot. At this central hub, we also collect precipitation data and other meteorological data. Click. As you can see, these little orange dots along the hydrograph, each one of these dots is a taken photograph that can be viewed by tracing the hydrograph or a step-by-step process, which is clearly defined in the full HIVIS page. Click. Click. After the water has meandered through the stream, we reach the outlet, which is comprised of 2 parts, as Derek said, the agri-drain and the overflow control structure. Click. On the HIVIS page, you can view the imagery in a video flip, choosing your own date range. All wetland sites are on this main page, and other sites across the USGS are on there as well. Click. And click. So there's the playback feature. Click. Okay. Over the last 3 years, I've made over 30 visits to the wetland per year for sample collection, measurements, and to verify discharge equipment and check the stability of the gauge. Click. The USGS has analyzed on average 70 samples at the inlet or the pump area and 75 samples at the outlet. Click. Using the hydrograph, determinations on what samples should be the most representative of the pumped water are sent to the lab for analysis. Click in the bottom left. No, sorry. Anyway, I was just going to say you can see how proficient our sampling is by the total flow of the inflow and outflow. So these percents here on just how well we're getting the coverage of the runoff. Next up.
Yeah, thanks, Chad. So I'll briefly cover the 2022 through 2024 wetland operations corresponding to the sampling that Chad just described, and then we'll really get into some more results here shortly. So typically we operate the pumps about 10 months out of the year, starting in mid-February and going through mid-December, and then we'll winterize the site for the coldest couple of months of the year when conditions are frozen. So we deploy our water quality monitoring sensors around early March after the risk of a deep freeze has passed. And so that, again, is the nutrient analyzers, ISCOs, and water quality sondes. And that's just because they don't have the luxury of a heated building. But also what we'll look into is some of the pumping statistics by month for each of the last 3 years with this slide here. So as you can see in the bar chart on the bottom, we have typical conditions for this area of the country or this area of the Great Lakes where we have wet winters in spring followed by drier summers and fall months. And that's what we see with our last 3 years of pumping at the Defiance wetland. In 2024, shown in yellow, we actually pumped substantially more water through the wetland than we did in 2022 and 2023. And that's partly because we decided to operate the wetland beginning in late January of that year due to a very mild winter. We really just had like a week of very cold conditions in mid-January, and then things warmed up for the duration of the winter. But also it was much wetter in 2024 compared to the previous 2 years, which had more drought conditions in the late spring and summer of those years. We'll go ahead to the next slide. Okay, so jumping into some of the results that Limnotech has generated with our sampling. First, we have results from the auto samplers, which what we do here is target event-based sampling at the internal wetland points, so at the end of cells 1, 2, and 3, and then we complement that with the monitoring that Chad and USGS are doing at the inlet and outlet, or cell 4 as it's labeled here. So we don't sample with these on our end throughout the entire year, but instead target the largest 3 to 4 events per year. Sometimes we get 5 events in a year when we're pumping for on the order of 48 hours or more before the pumps shut off. And what we try to do is stagger the sampling intervals with the ISCOs to last the entire event duration, and then use turbidity results to decide which subset of those samples to have analyzed. And we do that where we try to follow the same volume of water as it pulses through the wetlands, so that it's a fair representation of how water quality is changing with a certain slug of water that's pumped through the wetland. And overall, as you would see here in these 3 example events for 2022, 2023, and 2024 in the bottom, is for any paired sample, so sample 1, 2, 3, and 4 represents that same volume of water moving through the wetland. We'll see these incremental and gradual reductions in TP concentrations throughout the wetland system. So for any paired sample representing that same volume of water, there's a slight reduction in concentrations as you move from inlet to cell 1, 2, 3, and 4. Another observation that we've seen is over the course of an event, the concentrations entering the system continue to drop typically. So the first couple of samples will generally have the highest concentrations corresponding to that first flush of the runoff event. And then as you move into a few days worth of runoff, the concentrations will drop. And that's shown really nicely in the middle plot there, the March 3rd through 5th, 2023 event, with sample 5 having much lower TP concentrations in the system than the first sample. We'll go ahead to the next slide, please. Okay, so we also have turbidity sensors at the end of each cell that report measurements every 10 minutes to provide another piece of information regarding the water quality performance of the wetland system. And these are accomplished with multi-parameter sondes that also measure conductivity and water temperature. And here I just have a few examples of time series plots for some of the more significant events that we've seen, one for June of 2022 on the left, and then March of 2024 on the bottom right. And I'm just highlighting in blue the turbidity at the end of cell 1, and in green the turbidity at the end of cell 3. So really this is actually just about half of the wetland system. The changes in turbidity from that happen within cells 2 and 3, so kind of the middle two cells of the wetland system. And as you can see, this is generally the consistent behavior that we see from storm to storm, is that turbidity concentrations in the first cell are generally much higher. And then even within half of the wetland system, we can see a reduction of on the order of 50% in the peak turbidity concentrations, if you will, as that dirtiest water enters the system and moves its way through. And then as we continue to pump over the course of several days, these lines will start to merge as turbidity is dropping and it's finer and finer material. The turbidity lines start to merge, if you will, meaning that we're not removing as much of those solids later into the event, as it's finer material that would take longer residence times for it to settle. We'll go ahead to the next slide. Okay, so lastly is a look at our nutrient analyzer results. So here what we're doing is taking high frequency dissolved phosphorus measurements with online nutrient analyzers that sample. Now it's back to the wetland inlet and outlet at a duration anywhere from 2 to 4 hours typically. And we can see this data in real time as sample results are processed, which is neat for dissolved phosphorus. So these sensors we deployed during the March through May period. They're not deployed as long as the turbidity sensors or the ISCOs, but it's really during that critical months of the year when it's the wettest out there. And in the bottom now we have cumulative frequency plots for each year, 2022 through 2024. Blue again represents earlier in the system, the wetland inlet, and green is the wetland outlet. So this is a look at the entire season instead of just individual events to see how much lower the outlet concentrations are in green compared to the wetland inlet concentrations that are measured during all of these pumping events. And so big picture overall, we're seeing dissolved phosphorus concentration reductions on the order of 50% or more most of the time. And really what we see is greater reductions that correspond to larger, the hydraulic residence time being longer, which is for the shorter pumping events. And so just a little more on that note, I added this time series plot of March of 2025, so just last month, to look at dissolved phosphorus concentrations in a time series format, again at the inlet and outlet. And then we have the shading indicating the pumping events. So we had 3 almost 2-day pumping events in March of 2025. And then in between these larger events, you see these smaller shades, which represent the pulsing of water through the wetland. And so what we see as we pump longer and longer, the residence time gets lower and lower, meaning we're reaching a steady state condition. And during those conditions, we start to see the outlet concentrations get higher and higher, closer to the inlet concentrations, meaning the shorter the residence time, the slightly lower reductions in dissolved phosphorus concentrations. And then we get into the inner event pulsing pumping that's going on. We see those lines separate, again, more longer residence times, larger reductions in dissolved phosphorus concentrations is what we're seeing. So some really neat results so far, but again, very positive story to see the relatively good dissolved phosphorus reduction. And with that, I think I hand it off back to Chad.
So I'm going to go back to a little more broad view again. In this slide, I want to go through a snapshot of the year by year and briefly explain how and why some of these numbers vary. In this table, the thing to note is the timing of the runoff, which Derek kind of touched on, when the pumps were first turned on. In that first year, we had a lot of winter runoff when the pumps were not on. And in year 2, we got a little earlier start there in late February. And then in the third year, it was pretty warm last year, if you guys remember, and we were able to start pumping as early as January 8th. By doing this, we got to pull a lot more water from precipitation that usually happens in the February to March period. Next slide, please, or next click, I guess. There you go. At the wetland, as I mentioned earlier, we have a precipitation gauge, and each year we've seen a little less precipitation, but the timing of it has been different, as Derek alluded to. In the first year, it was pretty early. In 2023, we had some precipitation later in the season, but it was so dry by that time it didn't really register as runoff. And last year, we had very little very little precipitation even in the later months. One thing to note is most of this runoff came through in a couple of extreme large events. That's when we're seeing here in the winter time. Because of this, each year we've had an increased amount of water treated from Colwell Creek. As Derek alluded to, we and Shaylin mentioned this too, we doubled the pumps in 2024. So we were able to process twice as much water in that time, even with the effects of precipitation being a little varied in the wetland establishment. Changed the water quality components too, which we'll get to look at in this final table. Click. For runoff in early 2022, vegetation was just first getting established, so holding times were short and evapotranspiration was mostly non-existent. In year 2, we had much better reductions, and in year 3, we doubled the pumping and still maintained a 20% reduction in runoff. Click. For suspended sediment, along the same reasoning, no established vegetation early in the year, and most of that water came through the system, in the year, and when most of that water came through the system was positive. Even with doubling the amount pumped, wetlands still increased reductions from 2023 to 2024. Click. For dissolved reactive phosphorus, each year the percent reduced has lessened. I think that can be attributed to when plants are getting established, they take up a lot of dissolved reactive phosphorus each year because the wetland's been a little more established. There's been less new, but we're still getting the dormant season and then the grass coming back. So early in the year, so when we pump, we're talking March through June, we see most of our DRP collected, but in the later part of the season when new plant life isn't growing, what little runoff we do see, DRP actually increases. So that crucial period for reducing DRP seems to be coming in the spring in this particular wetland. And finally, click. TP has remained mostly steady through the first 3 years of this study. Next slide. For those who don't like tables, I have this graph here just so you can kind of see the same information. But what I want to draw your attention to are actually these sample bottles here on the left. So this first collection is from Colwell Creek. The second one's from the inflow, and visually it's like hard to see the difference. They both look pretty turbid, but the thing I want you to see is the outflow. If you look at the outflow bottles compared to like the inflow bottles, you can even visually see how well the wetland does, which I think is kind of comforting. Yeah, we can get the analysis done, but we can actually see the difference. It's pretty neat. Next. The last thing I want to highlight are just some pictures. I was asked to kind of notate the wildlife that visits the wetlands. So I've got a collection of pictures, 5 slides. I'll go through them pretty quick just to show you some of the different species that show up. But here we've got groundhogs. Some of them taking residence in these pumps. So here we've got raccoons that visit. Sometimes little fish get blown in through the pumps and the raccoon does cleanup duty. We got mallards here at the top. Next. Also at the inflow, we had a coyote. It's kind of at the right below the lettering. You can kind of see his backside leaving. I had a mink in the bottom left. Turkey and chick on the right side of the slide. Next. Last summer we had a family of turkeys, and throughout these pictures you can kind of see the progression of the little chicks growing up, especially in the bottom right. They're getting a little size to them there. Next. The inlet is our most popular area. We've got raccoons, we got deer, we got possum, We got a cat that lives here. I see him pretty much throughout the year, so I called him Tux. We got mallards, and finally, we've got turtle in the upper left, a belted kingfisher sitting on this wing wall, red-tailed hawk, great blue heron, and water snake in the middle pictures, and then I'm pretty sure these are otters in the upper right, unless someone wants to correct me, but it looked like a family of otters just based on the hand size and the eye spacing. And then the bottom left or bottom right, great blue heron. So pretty diverse group of wildlife. Pretty neat to go through the pictures and see them come and visit the wetland throughout the year. That's all I have. And next up is Derek again.
I think it's me.
Oh, sorry, Jalen.
Yeah, no worries. So, we've seen a lot of good stuff regarding like the water quality, all of this biodiversity, and some of these like large scale dynamics. What I want to highlight is part of our role as ERDIC. One of the things we do is we go out to the sites twice a year, and we sample at 60 locations within the wetland. And so, as part of that, over the last 4 years since the establishment to now, and we will continue until 2026, we've been tracking a variety of soil parameters, one of which are highlighted here on this slide, and that's total phosphorus. The total phosphorus is, to this point, this is up till September 2023, this first map here on the left. One of the things that you see is that, it's a really dynamic system. And one of the things that we really have elucidated from some of this initial TP data is that, I think there, if you click, I think there is a little thing that pops up. Yep. And so one of the things to note is that there's nowhere within the wetland that has zero phosphorus. So it goes down to about 100 milligrams per kilogram, but cool colors are high. These warm colors are lower concentrations of phosphorus. When we kind of zoom over to the right side, what we end up seeing is that the change in total phosphorus from its inception in 2021 till September 2023, it's really dynamic. These changes, I want to really point out that even though these numbers are varying across the wetland, and I think we'll see here in a second, they're varying probably due to a variety of reasons. If you click, we'll get one more pop up. But what you'll see is that, we get some areas where we're really, really retaining, some areas that look like we're retaining less. But it's important to note just that, inflow to outflow, we're really not leaching phosphorus back out into these waterways. And in fact, what we're doing is we're seeing the effects of the time it takes to ramp up and the processes that are associated with that. So, that's some of this phosphorus moving around. That's certainly vegetation establishing, using up some of the phosphorus, and the soil developing and continuing to bind more effectively. So next slide. As part of that, one of the other things that we are also monitoring for is SPSC, or the soil phosphorus storage capacity. The big takeaway on this, before we even get into the maps, is that to date, every time we've sampled at all points in the wetland, it has always stayed a sink of phosphorus, right? So there is nowhere within this wetland that is a source is now leaking phosphorus, as we've collected over 4 years, back into the waterways. And so this largest map here on the left, this is the change in SPSC from the creation of the wetland in 2021 till our most recent sampling period in September 2024. If you click, what you can see here is that these cool colors, this is where it's gained capacity. Maybe we have less P in that direct place over time. But then these warmer colors, these reds, that's lost capacity. So that's where we're really truly retaining phosphorus over time. And so up here in the right corner, I've got a a map that's showing the coefficient of variance for SPSC. Again, this is just another example of this system being so dynamic. So this is taking the variance at each sampling period in each year, and it is creating a coefficient by which we know how stable the system is with that phosphorus capacity. And, I can point out a couple of things here, right? Like, I know that some of these places where the variance is highest, we certainly are getting a lot more maybe inundation in those places. And as you'll see on this next slide, I think it corresponds well with some of the soil properties that are really driving the system. So next slide. And so what I am highlighting here are two soil properties and how they are potentially influencing our retention and capacity. So here on the left side, we have two graphs. One is total phosphorus, that's in the green, and the red is LOI, which is really organic matter. And so as we're increasing the organic matter, this correlates highly with increases in higher soil total phosphorus concentrations. The x-axis of these graphs is a profile. So along this black dashed line that's our channel. And what I've done is extrapolated points out across this line so that we can see how these parameters are correlating. If you click on it here, it'll highlight for us that, these peaks almost always correspond. So if we see high organic matter, or more importantly, if we see high TP, we're probably going to see high organic matter. A very similar situation is happening with our SPSC and our pH. The relationship between SPSC and pH, and if you click on it, we have essentially two zones here, which I think we consider kind of a lower SPSC and a higher SPSC. The lower SPSC correlates with a higher pH of the soil. This is likely due to the fact that, as pH becomes higher, it kind of goes in a scale, right? At first, it reduces the availability of these metal oxides. There's less for these phosphorus particles to really bind onto. And then as we increase the pH farther, right, then we start to get into this really strong relationship between solubility and pH that can result in things like precipitates out of the system. But the real key here is just that, like, these soil properties are really driving a lot of these internal variations, and they're really kind of the heart of the system. Next slide. And just quickly, the last thing I'll really talk about here is, as far as results go, is, we really try to utilize Defiance as a test bed for demonstrating emerging technologies and developing new methods for phosphorus capture. One of the ones that we have demonstrated recently, and this was in 2024, was our amendments. And so these amendments are lanthanum-doped iron particles. These lanthanum-doped iron particles, lanthanum is an element that is able to really strongly bind with phosphorus, and the capture on that is not only highly effective, it binds for a really, really long time. So we're talking more than 1,000 years potentially. And so what we do is we take these particles and we've put them in mesh bags, and we can deploy them, and we've tested them in both the mesocosms, and that's the graph you'll see here, and then we've also tested them at the Defiance site, and that's the upper right picture. This graph that you see, it's a little bit complicated, but on the y-axis, we have a cumulative rate of P retention, and then on the other y-axis to the right, this is the cumulative phosphorus retained, and that's at every time step. To get to the heart of this graph, what we really have found is that short-term deployment during highest P concentration flows is really where these amendment technologies excel. So for instance, this plot is showing bare soil plus amendments, and then just bare soil at the same pumping rate. This 4 to 8% relative removal efficiency is really sort of driven by the fact that bare soil is already highly effective. And so, if we think back to one of these first slides that I had presented on, bare soil at a low pumping rate has an effective removal of, somewhere between 86 and 90%. And so when we add 4 to 8% with these amendments, we're really getting to the point that the water is very clear, right? We're talking 95 to 97% effective. When we add in vegetation, not pictured here, over the course of the first 4 hours, we can even get up to like 20 to 30% increases in retention. And so these are going to need a little more refinement, but this is definitely a technology that could help us really drive long-term development and long-term management across wetland sites, not just this one, but others. Next slide. And so one of the final things I'll say here is, I just want to highlight that this group has been incredibly productive. It really has gotten a lot of really good guidance out, whether that's publications, attending conferences. If you see us at one, definitely come talk to us. We love talking about the project. We'd love to talk to you about anything you've got going on. We've had several outreach events. We're hoping to have more in the future. And then if you visit the website that Jacob had put in the chat, we will very soon have a brochure series online called, Practical Considerations for Wetland Design. And we go over a lot of the stuff we've talked about today in more depth in those that can be downloaded. And with that, I will turn it back over to Tim.
Good deal. Thanks, Shaylin. Yeah, I'll wrap up our last slide here, and then we'll take some time for questions as well. But just to kind of talk about our next steps and to give an acknowledgement, right? The work that we're doing in Defiance is, it's a highly manipulated wetland, right? You see, we have pumps out there, a lot of sampling equipment. It's not the type of wetland that we would expect, just any local stakeholder to go put in the ground, just given how built it is. But the reason that we did that, right, is to make it highly manipulative so we can really drive towards that optimized retention on the landscape. So acknowledgement that it's somewhat of a science project, but it does deliver its own sort of efficacy in terms of reducing nutrients. But part of our purpose is to really share that, right? To do that in forums like this, share what we're learning, and get it out there, get what we're learning out there within the total realm of wetland restoration within the Great Lakes and beyond. So to that point, just a couple future directions here, next steps that we want to, bring you guys in on, let you know about. For one is that we're looking at the opportunity to do a phase 2 of research, operational research out there in Defiance with support of the Great Lakes Restoration Initiative. So that's something we are in the planning phases for right now. That's something that would pick up after the current phase ends in May of 2026. So looking to define that opportunity and looking to do that within the context of existing research collaborations, right? We know there's a lot of other great work going on within Western Lake Erie, within the Great Lakes Basin nationwide, that we would really like to, further partner with any entity that's doing similar work in the region. That second point is that we are, specifically trying to translate this technology out into a more passive, less constructed type wetland. And we're specifically working an opportunity in St. Mary's, Ohio, in partnership with the Auglaize County Soil and Water District, with the city of St. Mary's, and with the state of Ohio through the H2 Ohio initiative, using one of our Army Corps cost share authorities to do surface water protection type work. That is a wetland that we are currently doing some environmental analysis and design for, that will be gravity fed, but employs the concepts of phosphorus optimization based on some sampling and what we already know about the site out there. So really exciting opportunity of how we can do this in a less sort of manipulated fashion. And then lastly, and something that we are really excited to talk about, is that we really want to get out there in the field with any interested stakeholder and spend a day, ask questions, and go down the rabbit hole of different interest groups to see how we can best share what we're learning out here and get that employed elsewhere in wetland restoration. We did that, I think, in April of 2022 or 2023, early on in the project. It was really beneficial. We've got some challenges on the federal travel side that is kind of throwing it up in the air. We were looking for a date in June that we would reach out to stakeholders like yourselves on the line with us and others locally to join us on site for a day. We are still looking for that opportunity. Little uncertain if June will work just due to some travel challenges that we have with the Army Corps and with the federal government at the moment. But we're looking for that opportunity, and I'll just leave it at that we want to find that space with you all and others to get out there. So with that, we got about 7 minutes, and I know our team might be able to stick on a little bit longer. I'm going to pause here and see what questions we can answer based on today's information. And Jacob, I know you're on the line. Maybe you've been looking at the chat. I see some things in there. Jacob, are you following any questions in the chat that would be good to jump into first?
Yeah, thanks, Tim, and thanks to all the presenters. There's a great question from Rick Wilson in the chat that I was going to direct towards Chad, related to the percent of the tributary flow that's being pumped. So, Chad, if you can take a look at that question in the chat. In the meantime, Santina Wortman had a great question about how long we anticipate this thing's going to continue to function. And while we're still working on the calculations for that, we haven't seen anything that would suggest that we're nearing kind of a phosphorus or nutrient saturation at the site. So my, initial response would be that, we anticipate this thing's going to continue to yield benefits for many years to come. More to come in terms of actually putting numbers on that, but so far we're not seeing that the absorption capacity for nutrients is being overwhelmed by any measure.
Yeah, Jacob. Let me get to Rick's question. Rick, in one of the tables I had that number. So in 2022, when the pumps didn't kick on for a while, and we had a lot of rain that came through in October through March 1st, there was 34 million cubic feet that went down Colwell Creek, and we pumped in 4.2 million. In 2023, there was 18 million that went through Colwell Creek. We pumped in another 4.4 million. Last year, there was about 18 million gallons or 18 million cubic feet that went down, and we pumped in 8.1. So that was kind of the point of that slide, was to accentuate like when our pumps went on. So by math, we missed, 88% the first year, 76 the second year, and 55% this past year. As Tim was alluding to, there's, this is very built. And because of that, we have restrictions with the pumps. We don't want to damage them. They're an expensive attribute that this site wouldn't function without. So we protect them, and because of that, we don't capture everything we could. But even in bare ground, as Shaylin has hinted at, or in fallow fields, we still see good reductions in, or see good efficacy in the wetland, even when it's not fully foliaged in what we think of as a beautiful wetland in the late spring, summertime. So I hope that answers your question.
Yeah, thanks, Chad. See a couple more questions popping up into the chat. Yeah, a couple comments as well. One from Steve: we could use the Western Lake Erie Basin Partnership to provide a tri-state interagency public type audience, which would be great. Steve, love that comment. Look forward to potentially coordinating that. Another question: do you monitor further downstream to demonstrate whether there is an overall reduction at the watershed scale? Chad, that might be a question best for the GS. Anything that feeds into a watershed scale that you're aware of?
I'm sorry, can you repeat that? I missed it. Do
no
you
worries.
also monitor further downstream to demonstrate whether there's an overall reaction? Oh, no. No, we don't. We can make those calculations. We know how much water's passing through. We know how much water's pumped in. I mean, we could say like the percents reduced from Colwell Creek But we don't actively have a gauge down there, just monitoring where the ditch that the outflow for ties back into Colwell Creek. Like I said, we could do the math, but we don't have something specific looking at that.
Yeah, thanks, Chad. And just looking at just a couple more minutes left, I just wanted to put one more plug into a couple great resources in the chat for those that are still with us. There is a project website. We are going to post this webinar in the chat or put into the project website once we get it up on YouTube. And then a plug too, if anyone on the line does not receive emails from us, if you want to be on our mailing list for things like events, things like this webinar, feel free to throw your name and email into the chat so we can catalog that.